Okay, great, thank you. So hi, my name is Alon Saguy. I'm from the Paninsky Lab at Columbia University, and I'm also an IBL member. Today I'm presenting a large-scale project of IBL named the Electrophysiological Atlas. The background for this project is that we're very interested in relating electrical signals to anatomical position in the brain to understand its structure and organization, and also its function. But it is very challenging to do so since there is very high variability in the electrophysiological signals we record using our probes. And there are many reasons for that. Different animals are in different states when when we record the neural activity. We use sometimes we use different hardware or different experimental setups. And you can see an example here from the reproducible electrophysiology paper of IBL, showing that different labs that targeted the same brain regions across different animals and tracked some electrophysiological feature, in this case, the baseline firing rate, they got pretty different values for their electrophysiological features caused by those variations between animal and different experimental setups. And the question we try to answer in 2025 is whether we are ready to develop universal models that would be able to explain the variability in electrophysiology across different animals and brain regions. To do so, we need to increase the amount of data we have across different animals and across different sessions. And since the early 90s and 2000s, where we probably mostly used a few units probes or focused on single animals or specific brain regions, we came a long way. When the Neuropixels were published in 2017, we were able to record from more than a single brain region simultaneously, allowing the addition of some context of the recording of a single neuron. So that was one important step in the development of universal models. Then Allen Institute's visual coding dataset, along with other datasets that started to be available at those years, were an additional step towards understanding how electrophysiology changes across different animals. And this project, along with others, have tried to enable us to generalize how electrophysiology should look like, and specifically here, it's in the visual areas of the brain. And then in 2023, IBL has initiated another large scale project that we in the EFIS Atlas sort of continuing. In this project, the brain-wide map, they used 700 neuropixels insertions to cover the entire brain, not in a very fine resolution. The resolution is, of course, varying as the density of the inserted probes change across different brain regions. But nevertheless, it was a very important attempt at understanding how the electrophysiology varying across the brain, across different brain regions, it might answer some question as whether we are able to detect that we are recording a certain brain region just by observing some electrophysiological features. But it weren't enough. And the goal in the project I'm going to present here is to maybe try to actually achieve that universal model that can explain electrophysiology across the brain. So here's our goals: to build a reference atlas of electrophysiology, to map neural signals to anatomical location, and maybe also genetic class. We also aim to provide some predictive models that would be useful for researchers trying to understand what they are recording at a specific insertion. And we would also like to share with the community some visualization tools that will enable you to understand if your recording contains some anomaly, if a certain insertion is actually targeting the wrong brain region, etc. So one way to go forward is by adding more insertions into the data set. In the IFIS atlas, we used 10,000, slightly more, which is not a significant increase in compared to the brain-wide map, which is contained in those 1,000 probe insertions. And as you can imagine, it still didn't help us to cover the entire brain in a sufficient resolution that you would expect from the electrophysiology atlas. So the remaining questions were, how do we fill those gaps, and how do we prevent anomalies in the recorded data from changing our model into a wrong a prediction of the distribution of electrophysiology in some regions. So the way we decided to overcome this is by incorporating additional information from other modalities. Specifically here, we're focusing on the AGEA atlas and the MERFISH atlas that were developed by the Alon Institute with some researchers also from Harvard University. Those are 3D reference atlases in the same spatial coordinate system, interpolated over multiple mice. They have different resolutions, so we upsampled the cell type atlas to voxel of 200 cubic microns. And you can see here an example of the gene expression atlas over 3 different slices of the brain. And each row here in this plot is a different slice. And specifically, we're not plotting here heat maps of a specific gene, but rather the principal components. Here you can see the 5 principal components of those of the genes in the gene expression atlas. What I want you to see here is that many genes or principal components of genes are very well localized in certain brain regions. And this is extremely important for us because we would like to add some information to our recorded electrophysiological data that will enable it to encode or a brain position, for example. So the way to use that information together with the electrophysiology is by recording electrophysiology in a certain position of the brain, detecting which genes were apparent at that position and what were their densities, also looking at the cell types around that position. And then when a similar pattern of cell types and genes appears somewhere else in the brain, maybe somewhere we didn't record before, we can probably predict that the same IFIS signature would appear there also. So in 2 slides, I'll show you some examples. But before that, I want to discuss which electrophysiological feature we are interested in. So we have 3 different groups: LFP groups, local field potential, marked here in green, AP group in purple, and waveform shape group marked in blue. Specifically, we decided to use those features since we believe they would be useful for other decoding tasks, such as brain region classification, probe localization, etc. You can see that some of them are more spread along the probe, and others are more local. Specifically, the waveform shape features are more local. And here are 3 different predicted heat maps of 3 different features across the brain. For the first time, we're aiming to present a brain-wide distribution of electrophysiological features. You can see one from each class that I've mentioned before, one from the LF, one from the waveform shape group, and one from the AP group. And what I would like you to appreciate is that, for example, for the LFP feature, the power spectral density delta, you can see a very high values in the hypothalamus, which is in line with what we know about the behavior of LFP in that brain region. So we are still working on validating all of those distributions and making sure that they are aligning with what we know biologically and anatomically about the brain. But I think this is at least from the first few weeks we've been analyzing those atlases, they look pretty much in line with what is currently known in the literature. Here's a summary table, more quantitative one, of the R-square score of all of the predicted electrophysiology features. You can see here that the LFP features are more predictable. We reach up to 70% of the variation in the data that could be explained by our model. Even though the waveform and the action potential features are less predictable, they are still useful for decoding tasks that I will show in a moment. And we're still working on improving the prediction of those features. Hopefully, when this work will be published, we'll be able to predict them even better. So here's the first example of decoding task where we will probably publish with this work. Here it's brain region classification based only on electrophysiological features. You can see from left to right, the Alon parcellation true labels, then the Cosmos parcellation true labels, and then the predicted labels of our model. The model is predicting the labels in the Cosmos parcellation in this case. You can see that it is not working perfectly, but it can predict pretty well the dominant brain regions in each channel of the probe. You can see that the confidence is very well detecting regions where we might have mistakes in the predictions due to some ambiguity in the electrophysiological features there. And the rightmost column is just the classifier predicted probability. If a certain row is dominated by a certain color, it means that the model has very high confidence that this is indeed the brain region that were recorded in that channel. And for a more comprehensive evaluation of the brain region classification, I'm showing the confusion matrix over all of the brain region in the Cosmos parcellation. We've been able to achieve 70% accuracy for the brain region classification, and probably there is still some room for improvement, especially in how we utilize the waveform and action potential features to improve that even further. Another application of the brain-wide electrophysiological feature models is real-time probe localization. It is extremely important in real time to be able to understand if we're recording the right brain regions, and also maybe to adjust the probe depth or prevent the probe from hitting the bottom skull. One way to do it is using comparing the recorded electrophysiological signals to the expected signals based on the IFIS atlas. You can see here an example on the left side of the slide. You can see a true position, XYZ position of each one of the channels of a certain insertion. This was estimated using a histology. Then you see the targeted trajectory in blue. This is where the experimenter planned to insert the probe. And in green, you can see the predicted trajectory, which in the majority of the cases is better than the target trajectory in means of distance from the true actual recorded positions in the brain. And on the right side of the slide, you can see the inferred brain region the probe was passing through. They were inferred by observing the XYZ coordinates. And I hope you can see that the predicted, the prediction of our model was able to detect which brain region we recorded slightly better than the planned trajectory. So to summarize, we have been working on the connection between electrophysiological signature and anatomy, and the two decoding models I've just shown you, the real-time brain region classification and probe localization prediction should be a very useful tool for researchers, and I hope we would be able to deliver those with very high performance and very few false negatives. We also enable the prediction of electrophysiology across the entire brain even in unrecorded regions. From the initial validations we've been conducting, the predictions look in line with the known literature. And hopefully in the future, this reference atlas could be serve as a healthy data set in order to enable researchers to detect anomalies in their data due to hardware malfunctioning or due to neurological disorders or diseases in a certain animal. I would like to thank the neurophysiologists that collected the data, and also to the IBL members, specifically those marked in red, and I would be happy to answer your questions.